Good morning, students. This is the continuation of the first chapter. The beginning we have discussed about the asexual reproduction, and in this asexual reproduction we have discussed about the different methods of vegetative propagation. Their advantages, disadvantages, everything we have discussed. Now next part we are going to start about the sexual reproduction. Sexual reproduction, important thing, involves two processes: the production and fusion of male and female gametes. So production of gamete, first method, and the second method is called the fusion of gametes. So the production of gamete is called the gametogenesis. Already the meaning we know. Gametogenesis, genesis means the process of production. Gamete. This process of production of gametes is called gametogenesis, and the fusion of gametes, male and female gametes, that means the later it is a process of fertilization. So sexual reproduction involves two steps. What are the two steps? Gametogenesis and fertilization. Now we will discuss about the they reproduce by the production of gametes. In sexual reproduction in algae and bryophytes, they also reproduce by sexual methods. They also produce the gametes, but in bryophytes and algae, their gametes may be motile, may be non-motile. What do you mean by motile? Motile means they can move from one place to another place. Non-motile means they cannot move, depending upon the species. So the gametic fusion is of three types on the basis of the nature of the gametes. It may be isogamy, when the two gametes are same in both morphology and physiology. Anisogamy, they are morphologically same but physiologically they are different. Oogamy is a type of anisogamy where both the gametes, both morphologically and physiologically different. In algae, the fertilization is external. External fertilization takes place, but in higher plants we can see the internal fertilization. So again one more question, where in which group of plants we can see the external fertilization? In algae, and higher plants, gymnosperms and angiosperms, we can see the internal fertilization. Now next part, this is all about the introduction of the sexual reproduction. Now next part we are going to see about the flower. Flower is considered as the reproductive part of a plant, and the flower is viewed in multi-dimensional perspectives from the time immemorial. The meaning of this sentence is, from long time back, the flower has been used by different groups of people in different ways. What are the different ways the flower had been used? Now, first one, it is an inspirational tool for the poets. First point, it is an inspirational tool for the poets. Poets, they are writing so many poems with the flowers. Number two, it is a decorative material for all the celebrations. Second way to use the flowers. Third one, in Tamil literature, the 5 lands are denoted by different flowers. So it is very much important in Tamil. The 5 different lands have been denoted by different flowers. Third is, the flags of some countries are embedded with flowers. And the fourth one is, flowers are used in the preparation of perfumes. So these are the 5 different ways flowers are used. The next part, we are coming to the biology. For the morphologist, a flower is a highly condensed shoot meant for reproduction. Already we know flower is a modified shoot. When the vegetative growth will be completed, the reproductive growth will be started, and the reproductive growth will be initiated by the formation of flower, and the flower will be produced either in the axillary position or in the terminal position. So that's why flower is considered as a modified shoot and is meant for reproduction. Already that one has been discussed in 11th standard unit 2. Now a flower possesses four holes. A typical flower possesses four holes. What are the calyx, corolla, androecium, gynoecium? Calyx, the unit of calyx is the sepal, the unit of corolla is the petal, the unit of androecium is the stamen, and the unit of gynoecium is the carpel. The process of changes involved in sexual reproduction of higher plants includes three stages. Changes involved in sexual reproduction. Don't be confused in the first part what I have told, that sexual reproduction involves two steps. Here we have, that is the gametogenesis and the fertilization. Here we are going to discuss about the changes involved in sexual reproduction of higher plants include three stages. What are the three stages? Pre-fertilization stage, first part. Second one is called fertilization. Third stage is called the post-fertilization. So before fertilization it is called the pre-fertilization. After fertilization it is called the post-fertilization stage. So there are three stages of changes can be seen in sexually reproducing plants. Now let us discuss all the three events in detail. So first one is called pre-fertilization structure and events. The hormonal and structural changes in plant takes place after vegetative growth. The hormonal changes and structural changes lead to the differentiation and the development of floral primordium. What do you mean by floral primordium? The flower development will be started due to changes in structure, and differentiation of the body parts also will be taking place, and the flower develops. The structures and the events involved in pre-fertilization are given below. First step is called the male reproductive part. We know the male reproductive part. It's called the androecium. Androecium is made up of stamens. Already we discussed, and just now we have told also. Each stamen possesses an anther and a filament. We know the structure. 11th standard we have discussed. Each and every stamen is having the two parts. What are the two parts? The bag-like structure which is present at the tip of the stalk is called the anther, and the anther is located on a stalk which is called the filament. So together, both anther and filament together it form the stamen. So androecium is made up of stamens. Each stamen possesses an anther and a filament. Anther bears the pollen grain which is inside this anther lobe. The pollen grains develop, which represent the male gametophyte. So one more question again, which is the male gametophyte? The pollen grains are considered as the male gametophyte. In this chapter we shall discuss the structure development of anther in detail. So next part we are going to discuss about the development of anther. A very young anther develops as a homogeneous mass of tissue surrounded by the epidermis. So with the diagram we will be discussing. Here the stepwise diagram has been given that we are going to discuss. So in the initial stage, when the anther development will be taking place, it is made up of, it appears like four-lobe structure, and it is made up of homogeneous tissue surrounded by the epidermis, epithelial cells. Next stage of development, just below the epidermis, certain hypodermal cells will be more prominent or conspicuous with conspicuous nucleus or conspicuous nuclei. Just below the epidermis, certain hypodermal cells will be very much prominent, and they will become enlarged with prominent nucleus. Second step, the next step we are going to say that these cells, the anther during its development, the anther assumes a four-lobe structure. In each lobe, a row or few row of hypodermal cells become enlarged with conspicuous nuclei. These conspicuous nuclei cells are called the archisporium. So here these cells are called the archisporium. Red color. You can see these cells are called the archisporial cells. Archisporial cells. Archisporial cells. Now these archisporial cells will divide. Archisporial cell divides anticlinally. Archisporial cells divide periclinal divisions. The archisporial cells will divide periclinally. Periclinally means, I will show here, the cells which are dividing parallel to that of the adjacent cells. Periclinal divisions. So that outside it will be developing the primary parietal cell, and the inner side of the anther it will be developing the primary sporogenous cells. So archisporial cell, they divide periclinally and produce outside primary parietal cells, and inner side towards the anther lobe it develops the sporogenous cells. These now one by one we will be discussing. The primary parietal cells which undergoes a series of periclinal and anticlinal divisions. After periclinal divisions, the cells will be dividing anticlinal, just perpendicular. So these primary parietal cells then divide a series of periclinal and anticlinal division and form 2 to 5 layers of anther walls composed of endothecium, middle layer and tapetum, that from periphery to the inside. So these cells primary parietal cells, their main function is to develop the anther wall. What are the 3 layers of the anther wall? Endothecium, middle layer and tapetum. Now next part we are going to discuss about the microsporogenesis. That means the sporogenous cell which has been produced from the archisporium, primary sporogenous cells that will be undergo changes in the formation of haploid microspores from the diploid microspore mother cell through meiosis. It is called microsporogenesis. The primary sporogenous cell may directly or may undergo few mitotic division to form the sporogenous tissue. This sporogenous tissue functions as microspore mother cell. Each microspore mother cell then divides meiotically and produce 4 haploid microspores. Microspores soon separate from each other and remain free in the anther locule and develop into pollen grains. The stages in the development of microsporangia already has been discussed here in the diagram. In some plants, all the microspores remain held together, called the pollinium. Example, calotropis. So one more question: what do you mean by pollinium? In which plant we can see calotropis? Pollinia are attached to a clamp or clip-like sticky structure which is called corpusculum. The filamentous or thread-like part arising from each pollinium is called the retinaculum. The whole structure looks like inverted Y-shaped structure which is called translator. So pollinium diagram will be like this: inverted Y, filament-like structure. This is the pollinium. So till here the development of anther. It may come for 5 marks question. The diagrams also to be practiced step wise. Okay, thank you children.